15 through 24. So I broke the groups up, and I'm going to start setting up breakout rooms in a few minutes. But because we had the exam and it sort of interrupted anything about potentially the anatomy of the digestive tract that you've been focusing in when you were doing your lab assignments, I want to remind you a few things that are going to become important today. And one of those is that the digestive tract and the stomach that we're focusing on today, it has a lot of ability to self-regulate through the myenteric and submucosal plexes that interlink a lot of parasympathetic ganglia that sit in the mesentery and are talking to each other, as well as being talked to by long reflexes that descend from the vagus. Meanwhile, you also have a sympathetic input that's coming from the ganglia that are along the spinal cord. And in general, sympathetic is going to be suppressive of motility and of secretions made by the digestive tract. So there's an interplay between these long reflexes, which are stimulatory when you are consuming food or anticipating consuming food, as we'll see, and the short reflexes. Now, there's a way in which those short reflexes can talk back to the long reflexes, and those are going to be referred in the book as the vagovagal reflexes, because sensory input coming from your digestive tract travels along the vagus nerve as it ascends. So that nerve, the vagus, is combined nerve carrying parasympathetic stimulatory signals to your ganglia in your GI tract, and sensation coming from the GI tract will ascend along that same nerve. We are also going to, hopefully, if we get through these phases, you're going to be starting to be introduced to a number of hormones that affect not only the stomach, but affect the entire alimentary canal. And those hormones are released by some of the simple columnar epithelial cells you're drawing that are down in the deepest part of the stomach or the intestine, called enteroendocrine. Entero meaning your digestive tract, endocrine meaning they release hormones. So these are hormones that sometimes act quite locally, and sometimes act great long distances, including interacting with your brain even. And they do it by being released into the blood. So I understand from some input that I've gotten from people who, thank goodness, listen to lectures ahead, that I need to work on one of the lectures that is posted. That's lecture 26. So what you'll see if you go to that lecture, it's on the liver and the pancreas, so it's something we do next week. It's just me talking, and the screen sharing apparently didn't happen. So whenever I was talking about a slide in the posted slides or writing on my tablet, you didn't see it. So I'm either going to investigate whether another version, which apparently exists in Zoom, does have all the stuff you need, or I'll re-record that lecture. So that's number 26 in your lecture number 26, and it's on the liver and the pancreas. So in a lab today, we are going to start talking about the liver and the pancreas. So our lab is going to go a little bit ahead because, of course, we have the lab test that happens during the same week as our class. It's a week from today? I'd have to look at the syllabus, but it's coming up, the 10th. I believe it is a week from today. So this is a lot to try and take in. So don't fall behind if at all possible. Go ahead and studying for the lab test. To help you with that, I spent much of the last office hour when I wasn't meeting with students, coming up with a flow chart to kind of synthesize what happens when you eat a particular macromolecule to its final destination, to the body cells or the liver, adipose tissues of your body. So I've tried to sew all of that together. That would be much of what's in the last essay, essay 6. So if we don't get to essay 6 in lecture, you're going to be learning some of this. Well, we'll get to it, but if we only do it very quickly, you'll have some stuff ahead of it, because the last lab will be looking at some aspects of this, that where are enzymes made, who makes them, what do they act on, what are their substrates, what are their products. And that lab is the one I referred to last time, which are the 3 videos, I think, from USF. That lab is next week on Tuesday, and there is a handout associated with it. And that handout is a new handout. If you were here last semester, it's not the same one. What it is you collecting the data from the experiment and then interpreting it. So if you ideally could do that lab beforehand, don't do the videos during the lab, but do them over the weekend, then we could really discuss the results so that you could turn in that handout and make sense of it before with more than 2 days to study it for the lab test. So this lab test is quite wide-ranging. I realize that. I'm going to try and keep it as straightforward as possible. But at times, what I might be trying to do is pull together things that are similar between, say, urinary and digestive systems. So you'll see the first topic today, the first breakout groups are going to be talking about one of the similarities, and that's the parietal cells. So let's start with breakout rooms, unless there are any questions. I don't see anything in chat yet. We've got about 2/3 of the class here, maybe a little. Well, we just lost somebody. So maybe a few people will enter, and I'll have to add them to your breakout rooms because we're missing about 10 or 11 folks. So let me get the breakout rooms going here. I'm going to, well, maybe I'll just let it assign automatically. That will speed things up. And so we'll create them. It just randomly puts you into things. Let me make the time 15 minutes again. Or maybe you only have 2 questions for each. So maybe I'll do it 10 minutes. Hopefully you're coming prepared and not, I know just the temptation is to take a break after the last test, but unfortunately there is no break until the end of the semester. So I'll give you 10 minutes, and then if it seems like you really need, I'll check in, you really need more than that, then we can extend that, or I can make new breakout rooms. I don't know what I'll do, but I don't think I can extend it. So let's go ahead into our breakout rooms. I'm going to rename them before I open them so you know which one you're in. Group A, and you're doing 15 and 16, and then rename them. B, you're doing 17 and 18. Name. Group C, you're doing 19 and 20. D, and you're doing 21, 20. 22, and the last group, E, you are doing 23 and 24. Okay, here you go. You have already drawn the gastric pits, and you hopefully have some idea, everybody has some idea of the order in which the cells are arranged within those pits. So who is the spokesperson to discuss why they're arranged in those pits? Maybe I will.
It's going to be me.
Okay.
Okay. So the surface cells come first, and those ones secrete a bicarbonate-rich mucus, and that's to counteract the acidity of the stomach acid. And the mucus next cells are the next layer, and they secrete an acidic mucus, which is to balance out the bicarbonate-rich mucus of the first layer. Next is the parietal cells. They secrete the HCl and intrinsic factor to regulate both of the previous two layers. The chief cells secrete pepsinogen, and that is that is to separate the H plus from the Cl minus.
Wait a minute. What?
That was
I think you just said it funny. You mean the HCl acts on that? The pepsinogen isn't splitting HCl. HCl spontaneously splits because if you stick HCl into water, it has a very high dissociation constant, which means that it almost entirely becomes H plus and Cl minus. And in fact, it's secreted that way, which is what we'll hear shortly. But instead, the HCl acts on the pepsinogen and forms what out of it? Is anybody in it?
Pepsin?
Pepsin, exactly. So once again, when you see the ogen at the end of something, just like angiotensinogen or fibrinogen, that means it's an inactive form, and you need something to cut something off to activate it. Well, in this case, it's the HCl. So that's good, and also pepsin can do it for itself. So there's a little positive feedback loop going on where once you make pepsin will split more of the pepsinogen and convert it into pepsin. Okay, and I'll let you keep going, Pradeep.
And then, okay, so that was chief cells. And then the last layer, the enteroendocrine, they release gastrin and hormones like histamine or serotonin into the blood into the blood to signal from the stomach.
Right, and there's a couple cells, let's go into the blood here. There are a couple cells that we're going to talk about, G cells and S cells. And these guys nicely release gastrin, because we're going to get to those in a second. Gastrin, sorry, I misspelled it. Talking and writing at the same time. And these guys release histamine. All right, so that is everybody. I didn't put the names of the cells, but what all the secretions are. Why do you suppose, and this doesn't have to be Pradeep, why do you suppose that they're in this order? Why do you want the enteroendocrine to be way down at the very base? What are they very close to then to get it into the blood? Starts with the L. Lamina propria. Now there is lamina propria all the way up here, but it's very thin. And there's certainly blood vessels up here. But if you look at the slides, as you get down to the base of the epithelial layers, you'll see a lot more lamina propria with a lot more blood vessels. So that's convenient to stick them down deep where there are more blood vessels, because they're going to be secreting into the blood vessels. When you're secreting into the lumen, the pepsinogen is secreted deeper than the HCl, and everything that's down in this lumen is going to be migrating out into the to meet the chyme out here. And as it goes up, it's going to encounter this acidic area. It's acidic partly because the mucus that's secreted is acidic, but also largely because of HCl being secreted, which will enable this pepsinogen to be converted into pepsin. Now, one of the themes you're going to hear is if it's a protease, and this is more for the lab today, if it's a protease, it has to be released in an inactive form. So these guys are always secreted in active forms. When you think about it, in order to secrete it, you have to use, these are, large molecules, so you have to use transport proteins in the membranes of these epithelial cells that are making the pepsinogen. And if it wasn't an active form, it would be digesting its own transporter. So by secreting it in an inactive form and then activating it outside in the lumen where either HCl is present to do it, and then eventually the chyme, the stuff you've eaten, the food, is there to act on. That way, hopefully, and we'll get to this in the next question, I think, hopefully you'll lower the amount of self-digestion that's going on. So this acidic mucus, they don't really give a reason, so I'm just kind of waving my hands here, but I'm assuming that this mucus is being secreted in an acidic form so that you don't neutralize too much of the HCl before it gets out into with the food that you've eaten. And then, of course, not of course, but we're going to talk next about some of the adaptations that help protect the stomach from self-digestion. So since this stuff that's on the surface of the stomach is encountering this mixture of food and secretions that have a pH of like 1 to 2, then you make an thick alkaline mucus to try and protect those surface cells. There's one other cell missing here, and that's that there are stem cells down here. They were missing in the figure, I think, too, in the book. They talk about them, but they don't mention them. So way down here, you have stem cells that are constantly generating these. And I actually don't know, but it seems to make sense that stem cells being mitotic here may push cells upwards along here, which means that a cell that was a enteroendocrine cell may, as it moves up, become a chief cell, which becomes then a parietal cell that becomes the mucous neck cell that then becomes the surface epithelia. I don't know that for a fact, but it's possible that that's what's going on. Or they could be making lots of different ones, and somehow they don't become active until they're pushed up into the part up here. So this is touching on a few ways in which the stomach is protecting itself against self-digestion, which is question 16. Are you doing that one too, Prabdeep?
I'll be doing it.
Okay, Jessica.
So there's 5 adaptations that the stomach has that protects itself against self-digestion. The first one is that the surface cell's mucus neutralizes the pH. The second one is that there are tight junctions between the epithelial cells, which regulates the movement of substances. And then The third one is that there's a rapid rate of mitosis every 3 days by the stem cells deep in the gland.
Okay.
And then the fourth one is endocrine regulation. So the stomach decreases the acid if pH drops below 2. And then the last one is that the stomach secretes pepsinogen, which is the inactive form of pepsin rather than pepsin. And as pepsinogen passes towards the lumen, it encounters HCl, which activates it. And once pepsin is present, it also catalyzes the conversion of pepsinogen to pepsin in a
Right.
positive feedback loop.
Okay. So your stomach is trying to keep a set point pH of around 2. And that's going to be one of the themes as we go forward. There's going to be some endocrine regulation that's going to try and maintain that. But it also speaks to a later question I think we have about the contents of the food and how it affects the pH of the stomach as it arrives, and then how that affects how long the food spends in the stomach. So we'll see that. That's a little foreshadowing. Okay, great, you guys. You did a good job in a short amount of time. So let's go on to 17 and the second group, Group B. And they're going to talk in particular about these parietal cells and the mechanism that allows them to release HCl. Now remember, this parietal cell, we'll let them go through it obviously, is basically an identical cell to a type A, except how it's regulated. So, so let's go ahead, and who is the spokesperson for the second group?
I can go ahead and answer that question.
That's you, Noah? Okay.
Yeah.
Let me just double check chat. Okay. We got an answer for something I missed in chat. I just didn't know if there's a question. Okay, go ahead, Noah.
So like you said, it's basically the same thing as a type A cell. On the basolateral side, it's going to be receiving CO2 from the blood, and that CO2 is going to enter the parietal cell and combine with H2O, and then it's going to utilize carbonic anhydrase to create carbonic acid. This carbonic acid will then split into bicarbonate and hydrogen, and then hydrogen is going to exit onto the apical side through an H plus, K plus ATPase antiporter. Also, while this is happening, K plus is leaving the parietal cell, and this is basically to fuel this antiporter to work. On the bicarbonate side, the bicarbonate's going to leave on the basolateral side through a bicarbonate chloride antiporter. And this is going to create like an alkaline tide as the bicarbonate enters the blood capillaries. And then the chloride will basically just, through facilitated diffusion, leave and go into the stomach's lumen. And then from there, it's going to combine with hydrogen ions to create HCl.
Okay, so why you need that potassium to leak out is so it can go back in. But you might recall from our electrolyte discussion last exam that some of this potassium that comes in also ends up going into the blood, because if you are vomiting excessively, that can lead to hyperkalemia, because your stomach's trying to maintain that pH of 2. When you vomit, you're losing HCl, so you make more HCl. The carbonic anhydrase is upregulated, and more potassium comes in, more of it leaks into the blood, and then you have a problem. So the chloride goes out in as a chloride shift, just like he mentioned. And once again, these two are facilitated diffusion. They're just moving along their concentration gradient. So if you didn't know any better, this is basically a type A cell. So the big difference is, and I feel sad that they got rid of this, is that these processes are all mediated by a secondary messenger system through cyclic A&P that you might recall. We're not going to go through the details of it at all, but as you increase cyclic A&P, that's going to cause this stuff to all increase. It's going to increase the rate at which this occurs. So what can trigger an increase in cyclic A&P there are receptors, on the, maybe I'll make them all look differently, on the basolateral membrane. And we're going to get to all the stimuli in a second, but acetylcholine is going to bind to some of these muscarinic receptors. And we're going to have gastrin, and we're going to have histamine. So all of these will independently cause an increase in cyclic A&P, and therefore increase the rate at which H plus is secreted and H plus and bicarbonate is made. So as he mentioned, there is an alkaline tide, which means that there's an increase in bicarbonate that is leaving the stomach area. And that blood eventually flows into the portal vein going to the liver. So that bicarbonate is going to be used, as you'll see later, by the liver, and for that matter, the duodenum and the pancreas also contribute. But the liver is going to be adding that to bile, because we're going to need it to eventually, in the duodenum, it's going to help neutralize the stomach acid. Because when we get into the duodenum, we need the pH to go back, acidic chyme in the duodenum. We need to bring that pH back down, or back up rather, to pH 7, because all the enzymes in the duodenum are going to need to work at a neutral pH. But in the stomach, pepsinogen, or the enzyme pepsin, is a rare enzyme who works best at a very acidic pH, a pH of 2. All right, so we did 17. 18 is a little more straightforward. There's nothing in the essays about 18, but it is just some facts to know. So what else is released is renin. Rennet, I thought it was. I think I misspelled that in the questions. Rennet, lipase, and intrinsic factor. So are you discussing 18 too, Noah, or is that something?
Yeah, I'll go ahead and do it too. Renin is an enzyme that digests the milk protein casein, whereas lipase will digest triglycerides. And then intrinsic factor allows for the absorption of vitamin B12 by the small intestine. So this can prevent pernicious anemia.
Oops. And intrinsic factor. So that intrinsic factor, we kind of skipped over, but it comes from the parietal cells as well, and it's secreted into the lumen. And we will be discussing this more when we get to the duodenum, but it allows you to absorb. It's required for you to absorb vitamin B12. So we did talk about this as part of, this brings up our review for the final, in the blood, because vitamin B12 is critical for mitosis. So in order to make blood used for DNA synthesis, in order to make blood at a high enough rate, you need to have enough vitamin B12. So folks without enough vitamin B12 can have something called pernicious anemia that we did talk about in when we did the topic on blood. Because of course red blood cells are being made at that whatever that godly billions of them a minute. So you need to do a lot of DNA synthesis in your bone marrow to keep your red blood supply up. So really, that's why I mentioned this before, but that's why they can dissect your stomach out in order to cause you to keep your weight down, except for the issue of vitamin B12. You need to have vitamin B12 supplements because you don't have the intrinsic factor to allow you to absorb it if you've lost all your stomach at least. As far as the rennet, I don't ask much about this. It's, there's some interest in that because eventually you don't digest milk as well if you don't continue to drink milk. So it is an inducible enzyme, and so you need the substrate, the milk, in order to the casein. Now, the other problematic one are lipids. So when we get to this handout that I've now put up on in Dropbox, you can get, you'll see that when they discuss this at the end of the chapter on how various macromolecules are broken down, you'll see that the saliva has lipase, the stomach makes lipase, and yet the textbook will say the vast, vast majority of lipids are not broken down until they get to the duodenum. So even in the lab today, or lab on Thursday, you'll see the little flow chart they generate says basically there's no breakdown. So why you make lipase, I don't know, in there. It doesn't do that much. Maybe it just gets mixed in enough that maybe it can act later. I don't know. But let's move on. So where we're going next is to start looking not just at the secretions, which, as I just pointed out, are both neurally through the acetylcholine and hormonally regulated, but motility of the stomach. So remember, the stomach has 3 layers of smooth muscle in their muscularis externae, and then it has a layer, the muscularis mucosa. So you need both of those to become more active so you can churn everything and cause mixing of all these secretions with the food that you've swallowed. But you also need to cause mechanical digestion by causing greater motility and eventually causing the food to leave the stomach and enter the duodenum. So who is going to do question 19 in the third group?
I'll be doing 19.
Okay, and I didn't see who you were, but
Kaylin.
Okay, Kaylin, go for it.
So the oblique layer of the muscularis externae in the stomach aids in grinding and peristalsis. The pacemaker cells, called the interstitial cells of Kegel, maintain slow cyclic waves of contraction. These peristaltic waves allow food to be moved from the fundus towards the pylorus. And grinding, or the most vigorous peristalsis, and most of the mixing action occurs close to the pylorus. Retropulsion occurs due to the pyloric sphincter closing during contraction. The pyloric end of the stomach acts as a pump that delivers small amounts of chyme into the duodenum as the pyloric sphincter slightly opens, simultaneously forcing most of its contained material backwards in the stomach. The pyloric sphincter prevents too much chyme from entering the duodenum at once so that the duodenum has time to process it.
That's very good. Okay, so just like in the esophagus, you get circular and longitudinal doing segmentation, squeezing, and longitudinal kind of waves moving along. All of this is coordinated through these sheets by having specialized pacemakers that called the interstitial cells of Cajal, and gap junctions between the smooth muscle in any one sheet of muscle. So all the longitudinal will send a signal of contraction that will go like a wave-like pattern down the length. And that would tend to move things up against that pyloric sphincter. The circular will squeeze and the oblique will grind for segmentation. Now, let's see what the next question asks. As she answered the next question already, describe the motions of the stomach and the role of the pyloric sphincter. But what we really need is going on to 20 is what are some of the stimuli that are going to cause all these guys to become more and more active, because that's what you're going to need in order to open that sphincter. You're going to have to, the stomach's going to have to get more and more active, almost in a positive feedback sort of manner, until at some point when the strength of contractions is enough, the circular muscles that form the sphincter will open during the crunching phase enough for chyme to leave the stomach. So that's question 20, some of these stimuli. Is that still you, Kaylin, or who's doing 20? Okay. Okay, so before we get there, the ACH is released on muscle layers, so you're going to increase motility. And what happens to the secretory cells with ACH? Oops. So they're also going to write, remember this over here where I put the ACH? I'm going to get rid of this thing so I can see it, the ACH here. You're just going to start to increase more HCl, but also more pepsinogen. So those are going to be stimulated by the ACH. Now, one of the things that I want to stress is kind of like in the ADH story of thirst response and high blood osmolarity, low blood volume. It's also not just what's released, but how much. So during the cephalic phase, you really only get a small increase in ACH. So that means you're going to get a small increase in motility and a small increase because you don't have any food there yet. Okay, I'm sorry. Go ahead, Stephanie. We're going to go feedback then, assuming that there's something there, there's going to be some food in your stomach. You're going to have some stimuli that feed back, right? On the vagus, that's where you were, I think. Okay, so when those stretch receptors, which are embedded in these muscle layers, when they get stretched, they're going to cause more ACH to be released. And that means you're going to get more motility. You're going to get more secretions. Now, that's good because you have food in there, and what that food's going to have done is neutralize some of this acid. So the pH is also going to be changing, and you're going to have chemoreceptors as well. So the one thing that we left out of cephalic is you could have the thought of food, but you could also have some other sensory inputs, right? So you could be right now thinking about lunch, just thinking about it, or you could have Yeah, you could have smelling. If somebody could be making it, you could have smell and taste, and those are obviously, you could see it. Yeah, you could say, wow, that looks really delicious. So all your other sensory come through. Now, eventually, and I think I'm going to end up doing this if we have time, because I didn't assign it. We'll talk about that little piece of the nutrition chapter, which is the regulation of food intake through the hunger center in your hypothalamus. So all of this is going to be going eventually through your hypothalamus to get to drive this vagus. Now the vagus, though, the neurons arise in the medulla, but we won't talk about more details of the brain than that. Okay, so are you doing gastric still? Or I believe the gastric is group D, right?
Yeah, I'll be doing the gastric.
Okay, and who is this?
Henry.
Okay, great. Henry.
So it has to do with the long and short reflexes. So the stomach will activate stretch receptors, which we talked about, which will activate the long reflexes via the medulla and vagus nerve. And then the short reflexes via the plexus, and that will increase ACH as well. And that both of them will increase stomach secretion, so HCl and pepsinogen, as well as motility. And then the food chemicals such as peptides and caffeine, as well as increased pH, will activate chemoreceptors, and that will cause G cells and H cells to secrete gastrin, histamine, and some other hormones into the blood, which will also increase stomach secretion and motility.
Okay, so pH, caffeine, and there's actually taste receptors. So you do have some sense in your stomach of, or in your GI tract generally, of what are the components in the food. But pH is for the stomach and things like caffeine. Did you mention something else, Manry? Receptors.
Peptides.
Put by a stretch here. Okay, so I'm putting these a little, she did a great job, but I'm putting these a little bit into two categories. There's long reflexes and there's short reflexes, which she mentioned. So the stretch reflexes act through a long reflex, that is the vagovagal reflex, that's these stretch receptors, to increase ACH. We talked about that a moment ago. So this cephalic phase is entirely long, right? It's all coming from outside the digestive tract. Once we get into the gastric phase, then we have a mixture of long and short that are happening simultaneously. And generally speaking, stretch receptors act through ACH and therefore have to act through long reflexes, vagovagal reflexes, versus chemoreceptors that act, and mechanoreceptors, I think they mentioned that too, but we'll just talk about the chemo. They're acting mostly through short reflexes. Now, that doesn't mean they don't increase ACH, but they don't do it by going out of the digestive tract. They're doing it through the ganglia. So you're going to see all of this increase ACH. And then did you mention hormones?
Yeah, I said the gastrin, histamine, and other hormones like serotonin as well.
Right. And serotonin. Okay, so we're starting into this morass of hormones. There is a table 23.1. Now you'll notice it's a big table, there's a lot on it. I would spend time with it. So some of the hormones we do discuss in more detail than others, like serotonin, we don't say very much about except here. But we do talk quite a bit about histamine and gastrin, and some of the ones later in the duodenum. So you do want to know that table. It's probably the most complicated part of this whole body system is the endocrine regulation of digestion. So gastrin and histamine, if we go back to this parietal cell, one of the things that you realize is not just how much acetylcholine, this is going to be increasing through those phases, but you're also going to start, at least in the gastric phase, to get more gastrin and more histamine. And what the book says is a kind of a good thing to picture, I guess, is if you just have your cephalic phase, you get a little trickle of HCl released. But once you end up in the gastric phase and the hormones are there, as well as, well, put serotonin since it wasn't mentioned, serotonin, as well as increasing motility quite a bit, it's like you turned on a fire hose from being a trickling, garden hose. And HCl plus pepsin, starts surging out in great amounts. So it's during that gastric phase, you get a lot more ACH, and you get, this all ends up being a great increase in both motility and in secretions. So your stomach starts churning harder and harder and harder, which is eventually going to not only mechanically break down the food, but ultimately open the pyloric sphincter a little bit and cause some of that food to leave and go into the intestinal phase. Okay, there are a few other hormones involved that we'll come back to later, but some of them are released in more of a circadian rhythm, and you know this because as lunch gets near, your stomach starts grumbling. One of them is motilin. It's got a great name. And because what it's going to do is increase motility in kind of a circadian rhythm way. So every certain number of hours you release more motilin, it surges up, and that makes your whole GI tract become more motile. So at this point, I think that's the only one we'll talk about. There's also another hormone released in the stomach that has to do with the hunger-satiety response, but we'll come back to that one later. Whoops, I have to leave this open. All right, so that is, let's see how we're doing on time. Oh, good, we've got hopefully lots of time. So we've gotten through some of all of Essay 1. So the very last sentence of Essay 1, which covers just really directly lecture questions 15, 16, 17, but it says what hormones and neurotransmitters affect the rate at which HCl is secreted. So that's what I talked about up here, and what they talked about during the phases. So that first Essay 1, you can just discuss these hormones, serotonin, and look in table 23.1, I'll just put it here again, to see if there's any other things you want to mention about that. So we've only talked about stimulatory. We haven't talked about inhibitory on this side of it. So given that it is parasympathetic that stimulates, what would inhibit would be an increase in sympathetic. So if you look at the figure that does these phases, you'll see on the right side there's an increase in sympathetic activity. This does release norepinephrine, but also through the way it affects the ganglia, it can release some other hormones that tend to suppress stomach motility or secretion, things like somatostatin. So take a look at that part. It's kind of on the right side. Now, under what conditions would those occur? Well, when you stimulate your sympathetic, it will tend to override your parasympathetic. So if you've ever been kind of hungry and then gone to the gym and really exercised, you'll notice that sensation of hunger passes as your sympathetic tends to suppress your parasympathetic pathways. But you also know that you could lose your appetite if you're ill or you're upset. And those things are activating sympathetic pathways that are going to generally suppress the feeling of being hungry. Although I know some of you, when you're stressed, eat and get more hungry, but there are different levels, I guess, of stress. And so that right part, I don't think I had I have it actually, I'm blabbing away on it, and it's actually question 22. So I'll let you guys develop any of these more when you cover 22. All right, so let's move on to questions 22, which is the situations that inhibit. So I just summarized them, but who was going to do that question? I'll put them down now.
I was gonna do that as well.
Okay, go ahead.
So there's loss of appetite with depression, and it will basically negatively stimulate the cerebral cortex, and that's going to decrease parasympathetic. So it's going to decrease ACH secretion, which will inhibit the stomach. And there's also excessive acidity, so pH less than 2, and that will negatively stimulate G cells. So there'll be less gastrin release, and thus decrease the ACL secretion. And also emotional stress, which will cause the sympathetic nervous system activation, which is going to override the parasympathetic system, and that's going to decrease ACL secretion as well. And all of these are abnormal except for the negative feedback.
Okay. Decrease secretions, also HCL, and this is a negative feedback. So the stomach is trying to maintain itself at around 2. For some reason, if you have excess acid produced, then you will release hormones in short reflexes. So this guy's a short reflex. These guys are long reflexes that will lower the amount of gastrin, lower the amount of HCl, and you'll make less acetylcholine, and you'll make less HCl. So that's by way of a short negative feedback loop. They don't go into much more detail than that. So that's all I would expect you to know, that you're trying to maintain that stomach around pH 2. Well, I shouldn't say that's all. I think it does say it is triggered with somatostatin, but that's okay. All right, there's one other big influence, which is question 23, and we'll put down number 4 here. I'm going to make a line, though, because this is now the intestinal phase. And there's a lot of hormones in the intestinal phase that are going to slow secretions and motility of the stomach in order to keep the amount of chyme arriving in the duodenum and the rest of the intestine small enough that it can do the major jobs of digestion that it has to do. So who is a spokesperson for Group E?
Hi, Professor. It's Jasmine. I'm just doing number 23, and I think we split it up into different spokespersons per question.
Okay.
So for 23, it is explain why the intestinal phase begins with a brief stimulatory effect, but quickly inhibits stomach secretion and motility. So I have 2 reasons why we have the initial chyme. It's the gastrin link to the chemoreceptors in the stomach to say that food has arrived. And it's stimulated again when it enters the intestines and allows it to move along and out of the anus so that there's room for food to enter into the digestive tract. And then the gastrin opens the valve between the small intestine and the large intestine. And then we have secretin, VIP, and other things from the intestine to tell the stomach to slow down, basically to inhibit, because you want chyme to come down, but in enough time to fully absorb before pushing new things into the jejunum.
Okay, good. So you kind of did 23 and 24 sort of all mixed together. Well, somewhat of 24, not all of it. But so as she said, there's gastrin released by the stomach, which is going to push the chyme into the intestine. But that initial, say you haven't eaten, you didn't have breakfast this morning, whatever. When you first eat, there's not much in your intestine anymore. It's moved onward. So when that first chyme arrives, you're going to have chemoreceptors that we'll talk about, including pH-sensitive ones, that will cause a brief release from intestinal enteroendocrine cells. And it's the same gastrin that you had released from the stomach. So initially, you're going to get a brief stimulation of the stomach. So you're going to get more secretions, more motility, which is going to cause more chyme to arrive. But that is very brief. Now, as she mentioned, the gastrin from the stomach, the gastrin from the intestine, it's all the same gastrin. It's just the source of it is one case stomach's G cells, and the other case enteroendocrine cells in the duodenum. It's all the same, and it's going to affect motility, not just of the stomach, but it does move things along in the rest of the intestine and ultimately down into the large intestine by opening the ileocecal valve. So that's great. All right. So for 24, some of this is changed by what's in the food, and that comes back to this negative feedback idea here. So are you doing that one as well, or is someone else doing 24?
Hi, Professor. I'm doing 24.
Okay, great.
So food contents allow for the activation of different receptors, such as chemoreceptors and stretch receptors, and this will enable the release of gut hormones, CCK, secretin, and as well as VIP. CCK activates via presence of fatty acids and proteins. Secretin activates via low levels of pH, and these hormones act to inhibit the stomach by decreasing motility and decreasing secretions.
Oops, that's not good. Oh, well. Cut them out. All right, so we're going to have a negative feedback on the stomach. Ah. Come on, thing. Okay, so that's part of it, and secretions. But the other thing is what's happening in the stomach, because what you're talking about, which I didn't go into, there's many hormones, you mentioned many of them. We're going to hear a lot more about those hormones. And they're doing that in a negative feedback in order to keep too much chyme from getting in the duodenum. But even in the stomach, how long things depends on what the food content is. So if you think about it, one of the ways in the blood that we neutralized acidic waste was we used the proteins in the plasma and the hemoglobin, and in intracellular fluid too. Proteins were buffers. Well, the same thing happens if you have a protein-rich diet. So if what you're bringing in is protein, it's going to tend to raise the pH more than if you have a carbohydrate-rich diet. It'll only raise the pH a smaller amount, increase the pH a lot, increase the pH just a small amount. So that's one reason why when you have protein in your diet, you tend to feel hungry more slowly the next time than if you have a pure carbohydrate-rich diet. Because when the food arrives, if it's all carbohydrate, then it's not going to tend to raise the pH much. You're not going to make as much stimulation of the HCl secretion and the pepsinogen secretion, and things can move along a little bit faster. If it's protein, you're going to tend to, you have those chemoreceptors in your stomach as well that detect that huge rise in pH. And through short reflexes, will keep the food in the stomach a longer time. And, that's kind of one of the ideas of having a very protein-rich diet for weight control. So we've started into what is one of the more complex parts of this, which are all the hormones. She mentioned a bunch of them, CCK, secretin, that are released by largely the duodenum in order to not only slow the stomach, but also stimulate the release of bile, of enzymes, and opening sphincters to allow those guys to get in. So we will be touching on that in the lab today because we're going to be looking at the liver and the pancreas and those sphincters. Let's see. So the question 25, I did not give a sign to anyone, but if you have your book and you open up I have it on page 888. It asks about, the question 25 asks about 5 stimulatory hormones that increase the stomach activity and 5 signals that inhibit the stomach activity. This is where you're going to find those answers. So if you look at the table, it has the hormone in the first column. Who produces it? Well, in this particular question 25, I'm not asking who produces it. It doesn't matter. I'm just saying, where is a source of a hormone that could increase? ACH is not a hormone, so don't use that as an example. That's a neurotransmitter. There are a few things that are sort of both neurotransmitters and hormones. For example, in that table, it says the stomach mucosa releases somatostatin. So some enteroendocrine cells are releasing somatostatin as a hormone, and neurons are releasing it as well. So if you look at the next column, it says stimulus that can cause its production. It'll say sympathetic nerve fibers. So I know it's a little bit hard to discern that from here, but generally take a look at the last column and say wherever you see inhibit or stimulates a stomach activity, then I would make that list. So this asks about 5, and what I have for 5 are gastrin, motilin, serotonin, histamine, and there's something called the substance P, which we'll discuss a little bit more later. So there's 5. Of those, the first 4, gastrin, motilin, serotonin, and histamine, are in this table. And so I would know those best. So perhaps I should have said 4 because I'm really not worrying too much about substance P. Now, of course, there's also acetylcholine that's released neurally. I would know that one. And then inhibiting it, as she mentioned, there's CCK and secretin. VIP, which is another one of these that is released largely by neurons. So it's kind of a neurotransmitter, but it is released into the blood, so it acts sort of endocrinely. And somatostatin, as I said, is kind of this mixture. So we have 5 there: CCK, secretin, VIP, somatostatin, and the last one is VIP. So we are going to be hitting all of this stuff on Tuesday when we start into the intestinal phase and the structures of the duodenum. So come prepared for that. Let's see, we have just a bit of time left. So I thought I'd spend a little bit of time, maybe I should find the picture, to share that picture on this last question, the hunger satiety response. I'm doing this in a really abbreviated sort of manner. Let me find the picture. I think it's in this one. And share that with you. This is in chapter 24 on the Unfortunately, right now it's in the cloud, so it's going to take a minute to open. On nutrition, metabolism, and energy balance. And what you'll see when we get to the liver as well, I am going to use parts of this chapter. Now, if you're going into nursing, at least you need to take a, whoops, I can't do that yet. You need to take a nutrition class, but I have been surprised that they don't do much on either of these topics. So that's why I'm taking parts of nutrition out of this, because I think it's important for understanding not only how you regulate food intake and fat levels, but also some of the many roles of the liver. So let's take a look in the slide, because I think the figure is quite confusing. This is the unedited version of it. I think in your lectures, use the edited version where I really cut things out. Unfortunately, well, I guess I can write on this. It's just not as easy to do the writing. So why I stuck this in here is both to understand that this is centered in the hypothalamus, but don't worry, this is the hypothalamus over here. Where's my writing thing? There it is. The hypothalamus here is where this is all being regulated. If you go back to 103A, that was one of the negative feedback functions of the hypothalamus, was the hunger satiety response. Now, if you look at this, it looks ungodly complicated. Don't worry about it. I'm not going to ask you anything about all of this. I'm not going to ask you anything about all of this. What I want you to focus in is whether you're feeling satisfied, your appetite is suppressed, or whether you're hungry. So there are different nuclei. Don't worry about who they are and how they work, talk to each other in these boxes. Just know that there are different nuclei in the hypothalamus. Yes, the brainstem is involved, but I never ask about that. So I'm trying to simplify this a whole lot. And you'll see along here familiar hormones or sensory activation, which is going to either cause you to feel more satisfied or feel more hungry. So let's go with the satiety. Hopefully it isn't surprising, or hopefully you listen to it, maybe not on Thanksgiving, but when your stomach becomes very distended and stretch receptors are saying, whoa, I'm really full, that is going to interact so that you feel a sense of satiety and decrease your hunger signals. If you have high levels of nutrients in your blood, glucose, amino acids, fatty acids, that's also going to say, okay, I don't need to eat very much. That means not only have you eaten and distended your stomach, but you've gotten food into your small intestine. It's been absorbed as these constituents, and that's going to end up being detected and send signals probably through chemoreceptors, from your GI tract up to your brain and say you're full as well. I don't need to eat so much. Now, there are some pure hormone signals, but those are not dissociated from eating. So as we get into the duodenum and the pancreas, you'll see that as you consume food, you're going to stimulate the release of insulin. One of the main ways that this is done is through GIP, but there are other pathways like just the absorption of glucose, which is probably the primary stimulus for rising blood glucose levels for insulin to be released. When insulin is released, it's going to also stimulate it. So it's kind of an echo chamber between the fact that glucose itself is suppressing, and insulin release is going to kind of supplement that and cause you to feel like you don't have to eat and you're satisfied. The other one is a little out of place here, but it has to do with maintaining body weight. So I'll try and write. I'm terrible with this thing. I'll try and write the word weight. So you have a kind of set point weight, which I'm sure you have experienced if you've tried to lose weight. It's very difficult because adipose tissue seems to have a certain amount of fat stored within it, and it tries to maintain that fat. And it does it through the release of a hormone called leptin, so that if you have plenty of fat in your fat cells, you're at your set point for their size, then leptin is released and it tends to stimulate a sense of satiety. You don't feel as hungry and you maintain your body weight. Okay, so these dashed lines are again inhibition. So you're seeing insulin will not only increase satiety, but it will actually decrease hunger. Well, what if you want to stimulate hunger? Well, that is through some hormones that we're going to mention. We mentioned in 103A over here, glucagon, which you might remember is when low blood glucose, that's your hunger hormone in a way, and it's going to go and stimulate the liver to release glucose, but it's also stimulating a sense of, I want to eat something, so you're going to feel hungry. Epinephrine, this is probably where your stress eating comes from, can also increase the release of glucose by your liver, but it can also make you feel hungry as well. And then there's another hormone called ghrelin, which actually stands for growth hormone releasing inducer or something like that. I've forgotten now. I think I say it in the lecture though. But that's where it was discovered. But now we know that this is released by the stomach, and it will stimulate it'll stimulate a hunger sensation and therefore wanting to eat. And it's released in a somewhat circadian manner too. So ghrelin is sort of the opposite of leptin. Ghrelin makes you feel hungry. Leptin makes you feel not hungry, satisfied. Okay, so they, for some reason unknown to me entirely, they mention insulin again here as a gut hormone. That's the same as this, that's just redundant. And then there is another hormone that is released to stimulate sense of satiety, CCK, and it's released, as you'll learn more about next time, when you have fat and protein in your diet. So that, again, is probably part of the idea behind weight control, where you have a higher intake of protein and fat. It's not only slowing exiting of stuff from your stomach and keeping your stomach feeling full longer, but possibly inducing CCK, which will then go through this pathway to make you feel satisfied. So that's a little different way of saying what I lectured in the lecture notes on this. And I just wanted to do it myself because of all these edits, that I didn't want you to be afraid of all the detail in this figure, and any detail that's in the text you can lower as well. Okay, so we've done really well. I managed to get through my ambition of all of these lecture questions. When we come back on Tuesday, we will enter into essay, the duodenum, and we'll sort of leave the stomach and not, because essay 3 talks about something we just addressed, explain how the contents of the chyme affect the rate at which it leaves the stomach. So you saw that kind of happen in 2 places. One, in the stomach, it tends to cause the pH to rise a lot more if there's protein in it. But then there's all these hormones that were mentioned as well. And we'll go much more into this. Really, essay 3 is where you're really focusing on table 23.1, although we certainly touch on it in essays 1 and 2. So we finished essays 1 and 2. We've touched on part of essay 3, believe it or not, already. And then essay 4 is again more of these same hormones, but we're also going into things you studied in 103A, which is the pancreas, the alpha and the beta cells. What are their targets? How do they affect the liver? That should be, I know it was a million years ago and a lot of water under the bridge, but it should be somewhat review. If you need to, look in the endocrine chapter to read a little bit more about that. So then my plan in the last couple of minutes, I'll just say, is essay 5 is really a part of this, again, chapter 24. And it's also a lot of review of the whole semester because, as you see, it touches on immune, the blood clotting, blood pressure. So it's a bit of a chance, and we'll be doing this on Tuesday on Thursday to start a review as a way of review. And then if you came in late, I mentioned this at the beginning, essay 6, I'm posted into Dropbox a flow chart that I'm hoping to help you with not only essay 6, but with the lab test. On Tuesday of next week, you're going to be doing a handout, which I believe is due that same day, or maybe I moved that. I don't remember. It may have been Thursday, which is collecting data from these videos of our lab exercise on digestive processes. So if you can do those videos, collect that data before Tuesday, then what we can do is go through essay 6 and at the same time the handout and explain the handout together with this flow chart I made in Dropbox. And that would finish up all our essays before the lab test occurs. Well, it won't finish up all of them, but we might even jump to essay 6 so that you have all that together for the lab test. I hope that makes sense. So I see something more in chat. Let's see what's in chat.
Professor, I had a quick question.
Yeah.
By Dropbox, do you mean like on Canvas, or do you have like an actual Dropbox link?
I have an actual Dropbox. I put a couple things in there. It seemed like other people found them. If not, I can try and...
Can you maybe put it in the announcements link?
Dropbox and put it in the announcements.
Yeah, thank you.
Yeah, I have the only way I can get these things I write on my tablet is into Dropbox easily, since they're big files. If I can, I'll share it. I'll transfer it into another more accessible thing if you have trouble with that. So just email me if you're having trouble getting it out of Dropbox. Okay, if there are any questions, we have about 5 minutes left. I know we came a long, long way today. Probably pretty overwhelming, so sorry about that, but time is short and we must move on, I guess. Okay, I have not started grading the exams. I will probably be doing things like your slides first because I want to make sure you get some feedback before the lab test. So I'll probably be working on that this afternoon, making sure you get whatever feedback you need for the lab test coming up soon, on a week from today. And that lab test will be, as always, I think I put this in announcements, it's going to be a PowerPoint that I share with you during the lecture time, which means that if we finished all of the essays or gotten close to them, then we could use the lab times on Thursday for review. If not, and we'll finish up the lecture on Thursday, but we have time the following Tuesday. So let me know if you have a conflict with our lecture hour on Tuesday morning during final exam week, if you've got a final scheduled. We'll try and find a time that most people can make it so we can have a review for both the comprehensive and the last midterm. So otherwise, I'm planning it to be during our normal lecture time on Tuesday. And then our exam, I believe, is Thursday at 8 in the morning, if I remember right, Lee. It's probably on my syllabus. I think it's 8 in the morning. It's not 9. It's, some ungodly. Yeah, 8 to 10 on the 17th, Thursday the 17th. All right, so if there are no questions, let me stop sharing this thing. Let me let you go, and we'll see you in lab. So in lab today, we're going to go through the liver and the pancreas. I showed you the models briefly. We'll talk more in detail about the models. And then we'll look at the liver and pancreatic slides, which is just a handful of those. And you can finish up all the remaining slide drawings. That'll be it. Okay. All right, guys. So if there's nothing else, I will see you in about 15 minutes in